introduce you to the fundamental sort of molecular biology that you need to be aware of in order to complete the bioinformatics practicals and to understand the theory behind what you're going to be doing over the next sort of few sessions. So these are the aims for this session, essentially to understand the biochemical structure of nucleic acids, so specifically DNA and RNA, and to understand the structure of amino acids and how they are translated into protein, and the structure of protein will be covered later on in another session. So there's a whole session dedicated to protein structure because it is, in its own right, quite complicated. We're going to look at how those sequences are represented in the literature and specifically for you on and in databases. We're going to look briefly at the flow of information through the central dogma. I'm going to elaborate on that through the processes of transcription and translation and link that to how genes, specifically eukaryotic, but I will touch briefly on prokaryotic, how they're expressed via those two processes. And finally, I'm just going to touch briefly on something that will become apparent later on, which is the concept of a reading frame and what can happen if that goes wrong will be covered in a later session. So, starting right at the beginning then, we're going to look at the structure of DNA, and hopefully you can remember that the structure of DNA sort of encompasses building blocks, so the kind of fundamental units of DNA and RNA are called nucleotides. And you can see a model up there. There's a model of the double helix top right in Cambridge. So if we were to look at the structure of a nucleotide, don't worry, you don't need to remember the chemical structure here. You just need to appreciate what you're looking at. You can notice that there are three parts to this. You have a phosphate group, if I get my laser pointer, phosphate group on the left-hand side here with a sugar. It's a 5-carbon sugar. So we start over here, carbon 1, 2, 3, 4, and 5, and that is therefore called a pentose sugar. It can be one of two types. It can be deoxyribose in the case of DNA and ribose in the case of RNA. There is a difference as to the structure between these two things. Hopefully you can appreciate, given the name deoxy before the ribose, hopefully you can appreciate what the difference may be. And again, I'll come to that later on. That is connected via a glycosidic bond to a base, and this can be one of the 4 bases in DNA or uracil in RNA. So in DNA, it can be adenine, thymine, guanine, or cytosine, and obviously uracil swaps in for thymine in RNA. In this case, it's guanine, and you can see the typical double ring structure of kind of guanine, or indeed adenine. They have both have double rings, whereas cytosine and thymine actually are only single rings, and uracil as well. Just to be wary that you should hopefully be aware that the phosphate group here is connected to the 5' carbon on the sugar, and the 3' carbon here links to the OH group, the hydroxyl group, which can be used to polymerize these nucleic acids into long chains to form a DNA strand. So essentially this nucleotide here, which is what we're looking at, has a structure. It goes from 5' to 3'. There's a polarity to it, and that becomes very important later on. That can be represented more simply in terms of kind of a diagram, which is probably what you're more used to. So there's a phosphate represented as the circle, the 5-carbon sugar here represented as a kind of pentagon. Don't forget the 5' carbon's actually here. There's an oxygen here, not a carbon. And then we just draw a line up for the glycosidic bond, and the base is just represented by a rectangle. So if we look at the structure of the bases, as I said, guanine and adenine are both purines. They are double ring structures. So adenine and guanine, you can see they're very similar, actually. They just have slightly different groups. For example, there's an amine group, an amino group there. Here we just have a ketone group. Anyway, the others are represented down here, cytosine, thymine, and uracil. They are single ring structures, and they represent the pyrimidines. So again, just be wary that they have different structures. They look quite sort of chemically, generally quite different. Apart from, some of you keen-eyed people might have noticed that thymine and uracil down here look actually quite similar. And actually, the only chemical difference is that on thymine, we have a methyl group sticking off here. But don't worry too much about that. So, because of their structure and because of their shapes, adenine can pair with thymine via 2 hydrogen bonds, and cytosine can pair with guanine using 3 hydrogen bonds. And again, hopefully you know that. But just by kind of looking at this, hopefully you can appreciate that it's to do with the structure of the bases. And the structure of each base determines how many hydrogen bonds they can form. So adenine and thymine have just the right shape, sort of conformation if you like, to permit 2 hydrogen bonding events between themselves. But either one can't really pair with cytosine or guanine, and vice versa for the GC base pair. Again, 3 hydrogen bonds here can be formed because of their shape and the chemical groups sticking off of their structures. This was determined by a guy called Shargaff, who died in 2002, but he was the one that essentially looked at the sort of percentage composition of bases in loads of different species and sort of organisms, and always found that there was a correlation between the amount of adenine and thymine and the amount of guanine and cytosine. However, the relative amounts could change and differ quite considerably between organisms. So archaea, for example, the organism I work on, tends to be very GC rich, so they don't have that many AT base pairs. Anyway, if you're interested in this, he published that in this paper here, and you feel free to read that. It's on the team's site, and you can read it in your own time. Get this one here. This one is a sort of perhaps sort of a secondary paper. It's more of a review than a kind of research paper. So if you want a bit more sort of information on Shargaff's kind of early life, his career, and sort of a bit of a watered down version, then feel free to read that one instead. So looking at polarity again, I've now numbered the carbons within the deoxyribose sugar here. So you can hopefully see that this is carbon 1 over here, carbon 2, carbon 3, carbon 4, carbon 5. Notice they are labelled with primes. That's just the way we do things, so don't worry too much about that. The important thing is to note that there is a directionality, there's a polarity to this molecule going 5' from the phosphate to 3' where we will connect. There should be a hydrogen there, connect to the next nucleotide. And you can see that again clearly here. And once we've connected 2 together, so if you look up here, we've connected 2 together, 5' to 3', and we form a phosphodiester bond here. So if you look at the double helix, you'll notice the bases stick out into the middle. The back, or sort of backbone as we call it, is actually made up of both the sugar and the phosphate groups. We call it a sugar-phosphate backbone, connected to each other via phosphodiester bonds. And again, you can see that here. So there's a kind of schematic on the right-hand side here. You can see the base pairs, again, 3 between G and C, 2 hydrogen bonds between A and T. And you can see the backbone in blue here, so that's the sugar and the phosphate going round, and the bases obviously stick out into the middle. Those 2 strands come together, wrap around each other in a plectonic coil, and you can see there that is now a double helix. You can see here on the left-hand side, this is what we call a space-filling model, and again, it just shows the same thing. You can see the 2 strands, one in pink, one in blue, running, and rather the bases running down the middle. So if you're interested, the double-stranded nature of DNA was sort of proposed and sort of published by Watson and Crick. So hopefully you are familiar with these 2 chaps. Again, the paper here, it's a pretty short paper. It's only sort of a page of A4, side of A4, so pretty easy reading. It's probably worth reading this. It's sort of one of those milestone papers in Nature. So again, that's on Teams. Please do give it a read. So we're going to talk about polarity a bit more. So as I mentioned, we can only add 3 nucleotides to the 3' hydroxyl group, okay, on the 3' carbon of the sugar. We cannot add a nucleotide to the phosphate group here, or the 5' carbon, and kind of polymerize in the other direction. Okay, we have to go 5' to 3'. We can only add to the 3' hydroxyl group. Now, all known polymerases, so both DNA polymerases and RNA polymerases, synthesize DNA in this direction, so 5' to 3'. You may ask yourself why. Why is there no known polymerases that polymerize 3' to 5'? And I will let you postulate on that one yourselves. If you are interested in that, I haven't got time to cover it, but do speak to me because it's quite an interesting reason, we think. So this sort of diagram on the right is just to show you what I mean. So this is RNA polymerase, so we're looking at transcription here, so generating an RNA transcript from a DNA template. And you can see that the enzyme here binds to this sort of transcription bubble and is kind of moving in this direction, 5' to 3'. RNA is being made. So again, that's just shown here, so that the RNA is being made 5' to 3'. We're just adding nucleotides sequentially onto the 3' end of the 3' hydroxy group on that sugar. And in the case of RNA, it will be ribose sugar. So let's just have a look a bit more detail in that. So I said we'd come back to this. This here is the structure of an RNA nucleotide. The reason is because the sugar is ribose, not deoxyribose. And the reason, or what I mean by that, is that deoxyribose on the 2' carbon doesn't have an oxygen for the hydroxyl group. It's just a hydrogen. So if you go back in your notes, you'll notice the DNA nucleotide purely has a hydrogen here, not an OH, not a hydroxyl group. Again, the reasons for that are quite important to the structural integrity of DNA versus RNA. And you may wonder why RNA has this if it's not very structurally sound to have a hydroxyl group here in terms of kind of chemistry and biochemistry of reactions. And actually, there are reasons as to why RNA wants to actually not be that stable. And you think about that in your own time. Hopefully, by the end of this course, you might actually come up with that reason. Again, speak to me if you're interested in that. But there are reasons for most things in biology. There's 99% of the time there is a reason that evolution has selected for these things. So as I said, RNA nucleotides contain ribose rather than deoxyribose. So just as a side note, deoxyribose is an abbreviation. It should actually be 2' deoxyribose because it's on the 2' carbon that the oxygen would be not present in DNA. Don't forget as well that thymine is substituted for uracil in DNA. So RNA molecules have uracil instead of thymine as a base. And again, there's only a minor difference between these two. It's only a methyl group. Again, you may want to either do your own research or speak to me if you're interested as to why thymine and uracil are interchangeable, and uracil is in RNA, but thymine is in DNA. It's single-stranded, don't forget. So RNA is single-stranded versus DNA, which is 2 strands that have come together via hydrogen bonding between the base pairs to form a duplex, a double helix. But just because RNA is single-stranded, as you can see by this image here, actually it can still form quite interesting structures and actually give off the impression that it's double-stranded in places. So you can see a hairpin here, where this strand has kind of looped back on itself and it's kind of base paired, certain bases along each bit of the strand. And actually you'd be fooled into thinking this was a double-stranded molecule, but it isn't. It is actually only a single strand. We're only looking at the top strand here, and you can see that actually it's just one strand. So don't be fooled by that. You can see again down here that another structure of RNA is something called a tRNA, a transfer RNA. We'll come across that with translation later on. Hopefully you've heard of these already. And again, they look like this. So this, believe it or not, is single-stranded. It's RNA. It just has a hairpin structure similar to this here because it actually folds back on itself and is quite an intricate structure. So again, it is technically not double-stranded as a molecule, but it can form what we call secondary structures, as you can see summarized here, similar to in tRNA. They call them secondary structures, hairpins are an example, but they can form loops, they can form bulges, etc. And again, it's always worth asking yourself, why is RNA single-stranded? Why is it not double-stranded? Why does it contain uracil and not thymine? Why ribose versus deoxyribose? Again, don't just take these as facts, think about them and come and speak to me if you're interested. It's always worth thinking about these things because biology doesn't do things randomly. Well, most things. Most things evolution has selected for in some way. Right, I'm going to speed up a little bit on this. So, hopefully you're aware that genes exist on chromosomes. They are bits of DNA that code for protein, or indeed regulatory RNAs. So, just because a gene is expressed doesn't mean it's translated from RNA into protein. And there are examples such as tRNA and rRNA, ribosomal RNA, that are not translated. Each gene consists of two strands because we're looking at DNA. And you'll notice of these two strands, there is one that's called the template strand and one that's called the coding strand. Only one of these is used for transcription into RNA. And it's actually the template strand that is used rather than the coding strand. And because it's the template strand that's used as the template for transcription, when each nucleotide base pairs with a template strand, we're actually generating this green strand here, we're actually generating a copy, if you like, of the coding strand. Apart from, of course, we're going to incorporate uracil. So the mRNA would essentially be the same as the coding strand, just with uracil instead of thymine. So looking at DNA sequence, or indeed RNA sequence, I've already talked about the fact that they have sort of intrinsic polarity, these nucleotides from 5' to 3', and that is actually how we report them in the literature. So if we just go back a slide, if you were to look at this gene here, we would actually, if we were reporting what the sequence was for this gene, we would only report, in the case of DNA, we would report the 5' to 3' strand. So whatever the sequence is, so A, T, A, C, G, G, whatever it is, we would report it going 5' to 3', not the other way around. So it's also worth just bearing in mind that genes actually can face in opposite directions along a chromosome. So as you can see here, we've got the chromosome with 3 genes, and what we're saying here is that actually genes 2 and 3 here are facing left to right, and gene 1 is facing right to left as we look at the screen. And obviously they are all still transcribed 5' to 3', as you'll notice. But they can face in opposite directions, and I'm going to talk a bit more about that now, or in a minute. So again, this is just an example of how we would report a DNA sequence and how they're stored on databases. 5' this end and 3' that end. Okay, and we just report that one strand because we don't need to report the sequence of the other strand, of the 3' to 5' strand, because you can work it out just by using the base pairing rules. So a little bit more about genes and their direction then. If we imagine this bit of DNA, and we've got a gene in purple here, it's going from 5' to 3' on this top strand here. And what's interesting is genes actually, you can almost think of genes as acting or sort of residing only on the coding strand of DNA. So only one strand, because the other strand is solely used as a template. So when we transcribe this particular gene, if it's facing left to right, 5' to 3' in this direction, the RNA polymerase will bind and it will move along the template strand, and we will then start adding nucleotides in a 5' to 3' direction. But it's worth bearing in mind in terms of gene directionality that if we were to take this exact same bit of DNA and flip it 180 degrees, it would look like this. In other words, the gene is now facing in the opposite direction. So it's all relative, essentially, as to which way round the genes are. Genes exists on the coding strands, but of course the coding strand going left to right here is the 5' to 3' strand, but of course we could have a gene going the opposite direction on the other strand facing right to left, and I've just illustrated that by flipping the whole thing round, so to hopefully kind of make that point sort of clear. And it's the same principle here. So all I've done is just flip the left-hand image over 180 degrees, so hopefully that kind of illustrates the point that it's all relative to which strand is which, and I'll make that clear in a minute. So as I said, both strands have 5' and 3' ends, and they therefore have essentially the same polarity. So both strands are kind of equal, I said in a guess. But because they're anti-parallel, genes therefore can face in alternative directions depending on which way you're looking at it. So just to illustrate the point here, if we have these two genes, we've got one facing left to right, one facing right to left. And again, they're all, this is just one sort of DNA molecule. And if we were to transcribe them, they would look like this, as we've shown before. But don't forget, which one is the template strand and the coding strand depends on which gene you're looking at and which direction it's being transcribed in. So if we were to look at the left-hand gene here, the sort of purple gene, the coding strand is where the gene is, here, and the template strand is at the bottom. But if we were to look at this gene here, then the coding strand is the bottom strand, in this case the 3' to 5' strand, and the template strand is the one at the top here, the 5' to 3' strand. So hopefully that's illustrated here. So essentially both strands, and this is the important bit, both strands can act as the template strand and the coding strand, depending on which gene we're looking at and which direction it's facing in. So looking at amino acid chemistry briefly, again, these are similar to nucleotides and DNA. They're the building blocks of proteins, and they have this sort of structure. There are 3 parts to an amino acid. We have an amino group, NH2. We have a carboxylic acid or a carboxyl group here. And then we have an R group sticking from the carbon. And finally, we have just a hydrogen. So we have a central carbon connected to both a carboxylic acid group and an amino group and an R group. And then there's just a hydrogen sticking off. The R group is variable. So what this actually is just a chemical group. It could be something like a hydrogen or a methyl group, and it could be something really complicated like a benzene ring. We'll get to the structures in a minute. Importantly, in the body, at kind of physiological pH, which is about pH 7, 7.4, we actually have this structure where it's a dipolar molecule, amino acid, called a zwitterion, and actually the amino group here has been protonated, so it becomes NH3+, and the carboxylic acid group has actually lost a proton, becoming O- here. So we actually have a positive charge on the left-hand side and a negative charge on the right-hand side, and that is now a dipolar molecule called a zwitterion. So you don't need to worry about this too much. I just wanted to make the point that actually in the body, this is generally speaking what amino acids look like. It's also worth just remembering, bearing in mind that amino acids do exist in three dimensions, so it's not just sort of two-dimensional on a page. You actually have these groups sticking out in kind of 3D, and you can then hopefully imagine that this R group, depending on how big it is, how charged it is, etc., can have quite sort of important kind of effects on the overall structure of the protein. So amino acids join together via condensation reactions. So as you can see here, water is liberated. So the two come together, the amino group of one amino acid, the carboxylic acid group of another, and we have a liberation of a hydrogen from the amino group and OH group from the carboxylic acid group from the other amino acid, and we get water therefore produced, and I've color coded it so you can see where those atoms come from. The resultant bond that's formed between the carboxylic acid end of one amino acid and the amino group of another is called the peptide bond, and for the chemists amongst you it's actually an amide bond, but don't worry too much about that. Importantly, what you'll notice is as a byproduct of the way that this reaction happens, we end up with two ends to this, in this case, these two amino acids, dipeptide. We have the amino group on one side and the carboxylic acid group on the other. So we call the amino side, or the amino end, the N-terminus, and the carboxylic acid group the C-terminus. And you'll notice that, again, like nucleotides, there's polarity going from N-terminus at the very beginning to the C-terminus, because don't forget, the next amino acid will come in here and will attach to the C-terminus here. So there is, again, an intrinsic polarity. And that's just illustrated here. So if we were to take this whole chain of amino acids with side chains represented, you can see that on this end we've got the NH, in this case 3+ group, so this is more accurate rather than NH2, but don't worry too much. And on the right hand side here we've got the carboxylic acid group, so again N-terminus to C-terminus. Looking more in depth at the R groups, it's the R groups that provide each amino acid with its chemical abilities, its character, which then overall dictates the tertiary structure of proteins and what shape they actually sort of fold into, and therefore what function they have. And again, as I said, it varies from hydrogen atoms, really small, tiny, single hydrogen atoms, to complex aromatic structures. So if we were to look at a simple example, glycine is probably the most simple amino acid. Again, we've got our central carbon, carboxylic acid group on one side, the amino group on another, hydrogen there, and our R group here is just a hydrogen atom. Okay, just a hydrogen atom in glycine, quite simple, quite small, quite unassuming. Contrast that with tryptophan, for example, which again has the same structure, apart from the R group is this massive sort of double ringed structure. It looks a bit like a sort of a base that we looked at, like a guanine or an adenine. Similar, but not quite. But this is quite a big structure, so hopefully you can imagine the difference that these two have. If you have a glycine and swap it in for a tryptophan, it's going to have quite a consequential impact on the sort of chemistry of the local area of that protein, and potentially therefore the function. And again, if we just look at that in 3D, you can see again the kind of massive structural change here to have this massive double ring structure versus just a hydrogen in glycine. So these are all the R groups, or most of them, that you need to be aware of. So again, glycine up here, the R groups are highlighted with a purple box. So it's just the hydrogen here. And again, if we were looked at tryptophan, that's what we saw just then. But you've got tyrosine, again, that's a ringed structure. Phenylalanine, you've got various charged groups actually. So you've got let me find the charged ones. Here we go. Lysine, you've got an NH3+ group on it. Arginine, again, it's positively charged, and histidine, positively charged. Again, you've got negatively charged, so aspartic acid or aspartate, you've got negative charge, and glutamate the same. So hopefully you can appreciate from this, you don't need to remember them particularly in terms of the sort of molecular structures, but just be aware that, you know, the R groups really do dictate function because they can vary in shape, size, hydrophobicity, how charged they are, and the reactions that they carry out. So just a quick sort of final note on proteins here. Just to be aware that the R groups do have an impact on obviously structure, but it also depends on where you find the protein you're talking about. So if we're talking about an aqueous solution, then the side chains, if they're non-polar amino acids, in other words, they sort of don't like water, they don't like dissolving in water, they're going to cluster together at the interior of the protein. So they're hydrophobic. If the protein is embedded in a membrane, it's an integral protein, for example, so like an ion channel, that kind of thing, then those polar amino acids in the side chains will move to this, or will be energetically more favourable to be on the surface of the protein to interact with the hydrophobic fatty acid tails within the phospholipid membrane. And that's just shown here. So again, non-polar amino acids are the interior of the protein here, with the polar on the outside if it's surrounded with water in the cytoplasm, for example. But if it's embedded within a membrane, you can see that actually all these yellow non-polar amino acids are sort of on the outside because they can interact with the fatty acid tails. So again, just to remind you of phospholipid structure, we have a phosphate head, which is hydrophilic. It's charged. Okay, it likes interacting with water. But the fatty acid tails on this phospholipid are obviously fatty. They're hydrophobic, so they exclude water, which is obviously the basis of the lipid bilayer here that is used for sort of cell membranes. So just to be aware as well, obviously water is excluded from this structure. It interacts with the phosphate heads, but not the fatty acid tails. So again, proteins have a polarity from N-terminus to C-terminus, and we therefore can represent each amino acid by either a single letter or a triple letter code, and we can write that N-terminus first going to C-terminus in the same fashion that we did with nucleic acids, where we went 5' to 3'. So again, this is just an example of single letter codes for amino acids, and I'll show you that now, in fact. So here are the single letter, one letter abbreviations and 3-letter abbreviations. So alanine you can write as ALA or A. Again, arginine, ARG or R. Some of them are a little bit weird, so something like glutamic acid or tyrosine, for example, is a Y. Some of them are a little bit odd, but hopefully they're sort of mostly sort of phonetic and easy to remember. But it's worth knowing the one-letter abbreviations for your computer practicals. So we're going to look at a little bit about protein folding. So don't forget there are 4 levels that you need to be aware of: primary, secondary, tertiary and quaternary structure. More details will be in Computer Lab 3. I'm not going to cover that now. However, it's just worth knowing, again, primary structure is this linear chain of amino acids. This will then fold locally into a select few structures, generally speaking, alpha helices and beta sheets, but there are more. But they are by far the most common, so they're the ones you need to know, certainly for your OCR exam board. And this is called secondary structure. And actually, I'll talk more about it later. It's not actually to do with R groups, it's to do with the nature of the peptide bond, but I will get to that later on. Once secondary structures are formed, you then get kind of more sort of broad folding into what we call tertiary structure, which is the overall 3-dimensional structure of the protein. And then tertiary structures can come together to form subunits of a sort of larger machine, if you like, that's called a quaternary structure. And actually most proteins in sort of your body, for example, are actually or do exist in some form of quaternary structure, whether that be a dimer or in this case a tetramer. It's worth just saying now that we cannot take yet the sort of primary structure of amino acids and predict accurately the structure of the protein that's going to be produced. Okay, even predicting secondary structures is quite tricky, but certainly predicting tertiary structures is still incredibly difficult and actually not very accurate, although there have been, as I'll talk about, some recent advances in this field, but it's still by no means concrete. So just to talk about the central dogma of molecular biology, this was first postulated by Francis Crick, and essentially what he said was that once information has been stored into a protein, once we've made protein from a gene, we can't then go back. It's a unidirectional flow of information. Again, this was sort of published here in the 1950s, so do feel free to have a look at that if you wish. This was interpreted by James Watson, again, Watson and Crick, the two chaps that came up with the double structure of DNA, double helix structure. And he actually kind of interpreted this statement and said that actually, well, DNA makes RNA makes protein. So this is generally what you'll see now as the central dogma, but actually it's not what Francis Crick originally said. If you want more detail on that, again, there's a slightly later paper here from the 1970s that Crick wrote. Feel free to read that if you're interested. But this is essentially what we mean. We have a gene as part of sort of DNA. During the process of transcription, we generate an RNA transcript using an RNA polymerase. Remember, it's made in a 5' to 3' direction because all polymerases work in a 5' to 3' direction. Why, you may be asking? Well, again, look it up or think about it. Come and speak to me. It's there are a couple of reasons that there's no sort of concrete answer to this, but there's certainly one theory that I think has more merit than the rest. Again, once we have this RNA transcript, this can be read by a ribosome and translated into a protein via protein synthesis. And again, the amino acids follow the sort of 5' to 3' structure because the 5' to 3' direction is then converted into a N-C terminal direction. So an amino terminus and a carboxyl terminus, N and C. The model of the central dogma saying that DNA makes RNA makes protein is actually sort of incorrect. It's very simplistic, I would say. And again, you'll come across this during your A-level studies. One of the reasons why this is deemed incorrect is because there is actually a way to go back from a transcribed RNA transcript back to DNA. And actually, a lot of viruses do this. So specifically, they're called retroviruses. And they will use an enzyme to carry out the process of reverse transcription, essentially going back from a single-stranded piece of RNA, mRNA, to a double-stranded piece of DNA, which is then called cDNA. Don't worry too much about it. We're not going to cover much about that during this course, but it's just worth knowing. So my analogy for this is that we have a sort of DNA molecule here, which let's just say is in French as the kind of genetic language here. This obviously can be repaired and replicated as normal during S phase of the cell cycle. This is then transcribed into a molecule of RNA, and all that is a copy of the template strand of this gene. So in this case, the gene is going from left to right. We have copied the template strand, which is the bottom strand here, and we've therefore produced a copy of what was the coding strand. It's just that it's a copy of that message rather than kind of using that message itself. So you can see, well, I've illustrated that there as if we've kind of taken a print from the DNA sheet there. Finally, we translate into protein, which then turns that French from the RNA into sort of a language, in this case English, that we can understand. And in terms of the cell, that's amino acids. And obviously linking them into proteins. So looking at transcription in a bit more detail. My pen's not working, here we go. Looking at transcription in more detail, we start with DNA and then we add RNA polymerase. We need some NTPs that are activated, so nucleotide triphosphates, and they're activated because they actually have 3 phosphate groups attached to them, which is required to provide the energy for this reaction to occur. There's also, quite importantly, a couple of magnesium ions that are required for this process to act as cofactors with the RNA polymerase, and the zinc as well, but we're not going to worry about zinc for now. And that gene, the template strand, or it's often called the minus strand or antisense strand, is then converted into an mRNA message of the plus strand or sense strand. So that's just a bit of terminology for you, don't worry too much. And this image here is just depicting actually some transcription. It's a snapshot of transcription that's happening in E. coli. So you can see there's lots and lots of mRNA molecules and sort of strands being produced there. And again, this on the left-hand side is just a sort of diagram of that process. So I'm just going to talk about two sort of additional bits just to kind of get your head around. And that is that there's a bit of an issue here that actually there are many genes within the genome. And if you are an RNA polymerase, you therefore need to know where your gene starts and where it stops. In other words, where do I start transcribing and where do I stop? Because what you don't want to do is transcribe every single gene, because energetically that's far too silly, it's not very favourable, and also you then can't regulate gene expression. The whole purpose of that being that you turn on selected genes and turn off others. So transcription is therefore defined to certain sort of regions of the genome that we know are genes, and they're turned on temporally. So they are regulated temporally, so only certain genes are turned on at certain times. So there therefore must be these start and stop signals for the RNA polymerase, and they are called promoters, which act as the start signal, and terminators, which act as the stop signal. So if we were to look at some DNA, here's the duplex, you can see the RNA polymerase is sat on top. It is sat on top of the region called the promoter, and that is the start point. It will then start transcribing along here, which you'll see in a minute, and it will then reach the stop site, which is the terminator, and it will then fall off. So just to make that clear to you, here we go. So the first thing that happens is that the RNA polymerase here, it has intrinsic helicase activity, so this will allow the melting and the displacement of the double helix in this region, and the breaking of the hydrogen bonds to allow access to the free nucleotides, to the template strand. And this sort of bubble here is just known as the transcription bubble. This happens at the promoter. So the RNA polymerase then works its way along the DNA, adding RNA nucleotides as it goes, forming phosphodiester bonds, because that's what the polymerase does. And as you can see, it just moves along, and it will keep doing so, producing the nascent RNA strand, 5' to 3', don't forget, in terms of direction, until it reaches the terminator, at which point it will dissociate, and the completed RNA transcript is then produced. And this process then starts again up top here for another gene, or indeed the same gene if it's being transcribed more than once. So, what determines which or sort of which direction a gene is facing? Hopefully you've now worked out it's actually the promoter. So that promoter sequence here has directionality to it. It tells the polymerase to bind to it and either start transcribing in this direction in the case of gene A, or in this direction in the case of gene B. So gene A in this case is living on this top strand here, and gene B is living on the bottom strand. But don't forget they're anti-parallel, so they're therefore facing opposite directions to each other. So hopefully you can sort of see that there. Don't forget, for gene A, the coding strand is the 5' to 3' strand, this top strand, and the template strand is this strand. But for gene B, it's reversed. So the coding strand is the bottom strand here, and the template strand is the top strand. But don't forget, the template strand is always 3' to 5', depending on which way you look at it. But it's always 3' to 5', and the coding strand is 5' to 3'. Don't forget, we only note down the coding strand. So again, this is just showing promoter for gene A is facing this direction, promoter for gene B is facing in that direction. And again, we only note down 5' to 3', like the coding strand for each gene in the literature. So once we have transcribed and we've got our molecule of pre-mRNA, we need to do a few things. Because don't forget, we have introns and exons in eukaryotic systems, and we therefore need to remove these introns from the coding sequence, and that is done via a process called splicing, where there's a kind of molecular machine that's made up of protein and RNA called a spliceosome that will come along. It can recognize these junctions. Again, how it does it is kind of, we're not going to worry about for now, but if you're interested, come and talk to me. And it will cut out, it will excise these introns and join the exons together to form one sort of molecule, one region, if you like, that's not interrupted by introns. Oops. Just be wary that this molecule called mRNA would now be translated, but not all genes are actually translated. A lot will now remain as RNAs, so tRNA, rRNA, and there are many other examples. So looking at translation then, it's carried out by the ribosome. Again, hopefully this is all sort of revision, so I'm sort of going to speed up a little bit. mRNA is made up of codons, and 3 bases of DNA make a codon in RNA, which is then read by the ribosome in sets of 3. Each codon is specific to a tRNA molecule, and each tRNA molecule carries a specific amino acid, hence how you can then determine what amino acids are sort of polymerized. The ribosome catalyzes the formation of the peptide bond between amino acids as it translocates along the mRNA molecule. And again, there are start and stop signals for this. So there's a start codon, specifically AUG, although I've put a star here because, again, most things in biology are not kind of 100% the law. And actually, there are alternative start codons in various organisms that are also used, but by and large, it's AUG as the start codon. Don't forget, this would be ATG in DNA. The stop signal, there are three stop codons, so UAA, UAG, UGA. And this is the reaction that's carried out by the ribosome. So you have these 2 amino acids, as I said, they get joined together via a condensation reaction, and a peptide bond is formed between them, as I showed you earlier on. Of note, just be wary that this reaction is indeed in equilibrium, but actually the reaction is kind of favoured here, as you can see by this arrow. It's far more favourable to be on the left-hand side of this reaction, so the substrates, the substrates, rather than the product. So actually, just be wary that we need the ribosome in in vivo, to carry this reaction out. Okay, if you just had these 2 amino acids in a test tube, they wouldn't really come together and form a dipeptide. You have to have the ribosome there to allow this reaction to occur, generally. And don't forget, as with everything, water's produced, so it's a condensation reaction because we're joining 2 things together and liberating a small molecule. In this case, it's water, but it's not always water, so just be wary of that. DNA polymerization is a condensation reaction, but water is not actually the leaving group in that case, so just something to note. So I'm just going to talk briefly and hopefully revise the structure of tRNA. So these molecules are the link between the codons in mRNA. Remember, that's three bases. And the specific amino acids that come together and are sort of polymerized together via the formation of peptide bonds. It's done via the use of anticodons in the tRNA that are specific to the codons in mRNA. So I'll show you the picture here. You can see that here we have our tRNA molecule. Remember, it's actually single-stranded. We just have these hairpins. And it has a 5' end here and a 3' end here. The 3' end is connected to the amino acid, in this case phenylalanine, but down here we have this anticodon. So you can imagine mRNA is kind of along here. The anticodon can pair up to a specific codon in mRNA, and then that allows this amino acid to be selected for. The next one will come in, and this amino acid will be attached, again via a peptide bond. And don't forget, it's done via this code, this genetic code, which we talked about last time. And that's that. So I'll just show you that picture in a bit more detail. So again, we've kind of flipped 180 degrees, sort of laterally, so just be wary of that. So the 3' end is now on the left-hand side, attached to methionine. And you can notice that we have our anticodon. Again, we're going to say it's 5' to 3', CAU. So 5' to 3' is going CAU. And again, we have the mRNA molecule here going 5' to 3', AUG. Okay, don't forget this is anti-parallel again. We have the 3' end of mRNA on the right-hand side, which is actually lining up with the 5' end of the tRNA. So it's anti-parallel again, because don't forget it's all nucleic acid at the end of the day. Right, we're nearly there. I'm just going to sort of touch briefly on what I've said about translation. So the initiation of translation happens if we have the mRNA molecule here at the bottom. We start with AUG, the canonical start codon, and this encodes for methionine. So the tRNA molecule, again, the example is here. So AUG in mRNA, we have UAC as the anticodon in this tRNA molecule, which is connected to a methionine amino acid. What will then happen is another tRNA molecule will enter the ribosome and will base pair with the next codon, which is GCG. Okay, so GCG, you can guess what the anticodon would be. So here we go. Comes along, it base pairs with GCG, so the anticodon on the tRNA molecule will base pair with, via hydrogen bonding, with the codon of GCG on the mRNA molecule. They are then close enough together for the ribosome to catalyze the formation of a peptide bond between the amino acid of the first tRNA molecule, which is methionine, and the amino acid that's coded for by GCG, which is alanine. So the bond will now form between these two, catalyzed by the ribosome. Hopefully you can see that here. Once that bond has formed, the ribosome will then translocate. So it will move 3 bases upstream, and that requires ATP hydrolysis to provide the energy for that to happen. It does actually also require GTP hydrolysis, but don't worry about that. And the tRNA that was currently in this site with alanine, therefore gets shifted into the next site in the ribosome. And the tRNA molecule that's now not connected to the methionine anymore, because don't forget methionine is connected to alanine, which is connected to this tRNA. This free tRNA that no longer has the methionine attached gets kicked out of this side and then recycled by the cell. Then the cycle continues. Okay, so we have another tRNA molecule coming in, this case arginine. So what's that? CGG is the codon that specifies that. And the same process will happen again, as you can see here. Just one note, if the tRNA molecule is connected to an amino acid, it is therefore referred to as a charged tRNA molecule. If it is no longer connected to an amino acid, it is uncharged. And actually an important point which you may think about is how does the cell know to put a methionine amino acid onto this specific tRNA and not the one that actually would bind here that should be an alanine. So that's just an interesting point that you may want to either look up or, again, speak to me about. It's a fascinating topic. It's something that you may not have considered before, the kind of charging of tRNA molecules, but actually it needs to happen. Okay, you as a cell need to attach the correct amino acids to the correct tRNA molecules so that your code will work. In other words, AUG is not recognized, is not bound by this tRNA that actually is connected to the wrong amino acid. In this case, maybe if it would be connected to an alanine or a phenylalanine or something, you actually need to make sure that you're connecting a methionine to the correct tRNA molecule. Again, a reminder of the reaction that occurs. It is a condensation reaction, and therefore water is liberated. The final point for this session is just to talk briefly about reading frames, more detail later on. But if you were to take a molecule of mRNA, as we see here, there are 3 potential reading frames. You could start at AUG and read AUG as the first codon. That would code for methionine. You then look at the next 3, AAA, and then GCA, AUU, etc. And you therefore get this potential sequence of amino acids. You could, however, read it starting at the U, so you could read UGA, and then AAG, CAA, UUU. And hopefully you can appreciate that that has actually changed the amino acids that have been coded for. So all the way along here, you've got a different set of amino acids. Finally, you could start at the G, so you could actually go for GAA, AGC, AAU, etc. It is actually the top one that works, because the start codon sets the reading frame as being this reading frame at the top here. Okay, so AUG sets the reading frame essentially. Otherwise, if I just gave you a random bit of sequence, you don't know what frame it's in. Okay, so if you ignore this AUG here on the left-hand side, imagine I just started with AAA. Who knows whether that actually starts AAA or whether it actually starts AAG or even AGC? Who knows? But of course, you do know that if you know where the start codon is, because you know the start codon will code from methionine, AUG, and therefore that will then set the reading frame downstream. The final point just to remember is location, about where these processes happen. So transcription happens in the nucleus. The mRNA is then transported out via a nuclear pore through the nuclear envelope, and then is translated in the cytoplasm where the ribosomes reside, or indeed actually in the rough endoplasmic reticulum potentially as well. So just being aware of that. So that's the end of this podcast, and obviously next week you'll be doing an introduction to the computer practical, so I'll be talking about databases and homology searching, and then you'll have a go at the practical the following session.